Okay, welcome to video number 46 of the Diaries of a Coronavirologist YouTube channel. Today is the 23rd of December, and we are up to almost 78.5 million confirmed cases of COVID-19 around the world, and about 1.7 million confirmed deaths. So today I want to talk a little bit about the UK variant that has recently emerged, or more accurately, the B.1.1.7 variant of the SARS-CoV-2 coronavirus that causes COVID-19. So I've broken this video down into three different main ideas, which I've timestamped below if you want to jump around things, but they are all tied in together. So I'm going to talk about virus mutation initially and natural selection as a consequence. Viruses mutate very rapidly and very commonly, so natural selection is super sped up for viruses. Within that discussion, I'm going to lay out an idea and a framework for thinking about this critically. So when you see that a virus mutant variant is now at a high prevalence within a population, that doesn't instantly mean you can say it's more transmissible. We still have to approach that with some degree of skepticism. However, in the case of the UK variant, it does seem like this virus is indeed transmitting faster, but that is only based so far on genetic and epidemiological data. We haven't got any indications from lab-based experiments about its ability to infect cells, for example, and I'll talk about all of that, while also talking about the mutations that we're seeing and what impacts they might be having on this new variant of the virus. I'll then end, of course, by talking about what impact this may have for immunity and the vaccines. So the spike protein has mutated in this variant, and that could be enhancing transmission. By extension, it could also impact people's immunity and vaccine-derived immunity because antibodies target the spike protein. However, so far, there's no reason to think that the vaccines and natural immunity will be impacted because there's no experimental data to show that. So that's just sort of where I'm going with all of this video. So this variant isn't a new strain of the virus, it's just a variant that's defined by the fact that it has a significant number of mutations that change it compared to some of the other circulating variants that are out there. So mutation in viruses is not uncommon. Viruses mutate very frequently and very rapidly, so it's not a surprise to see new mutated variants emerging. I've previously talked about this on the channel quite a long time ago, and I'll put a link down in the description of this video to my previous one where I went into mutation in a lot more detail. But to briefly say it here, mutations occur when the genome of the virus isn't copied perfectly and therefore changes. So new viruses that are produced from a cell that becomes infected are different to the virus that infected that cell because of genetic changes. Those genetic changes can be of two different types. They can either change the protein that the virus produces because they've changed the DNA in such a way that those instructions are significantly different and therefore the protein that comes out at the end is different, or the mutation can not impact the protein. So it's possible that the DNA can change, but the protein at the end stays exactly the same. So these two types of mutations, given their technical name, are known as non-synonymous and synonymous. So a non-synonymous change is when there's a change to the protein. A synonymous change is when there is a change to the DNA, but not to the protein. Now, as I say, mutations in viruses occur very frequently and very rapidly, and they can be of different consequences. So more often than not, they're going to be detrimental to the virus. So viruses mutate a lot, but a lot of the time that is actually going to be completely detrimental to the virus and kill it in essence. So if the new genome that's produced has mutations that stop the virus performing all the functions it needs to initiate a new infection, that virus is in essence dead, it's non-infectious. We never really see those viruses though because they can't go on and infect cells. So when you look at viruses that are circulating in a person, you're never going to see those detrimental mutations because they don't spread in a population. They get wiped out and extinguished. Through the process of natural selection, therefore, beneficial mutations can be selected for positively and can start to rise up within a population. As I say, with viruses, they mutate incredibly rapidly, so this natural selection is a lot faster than what Darwin would have been thinking about and defining. But the same processes of evolution that have led humans to evolve to what we are now from our prehistorical ancestry It's the same process going on in viruses. They are evolving and changing, and natural selection works on them just as it does any other living organism. So therefore, anything that has a positive impact for the virus's ability to infect its host, so in the case of SARS-2 that caused COVID-19, infecting humans, that can be positively selected for through the principles and processes of natural selection. Now I'll come back to increased transmissibility because that's what we're talking about here with the UK variant, but I also want to point out that there can be neutral selection. So viruses are constantly mutating, constantly changing, and they can have these non-synonymous mutations which cause the proteins to change their sequence and potentially their structure. But it's possible that that has neither a beneficial nor detrimental impact for the virus. So it doesn't actually have any selective advantage or disadvantage to the virus. It's just a mutation that's occurred and doesn't do anything. Now, why I bring up neutral selection is because the fact that we're seeing an increased incidence of a certain variant of the virus spreading doesn't instantly mean that it's more transmissible. There could be a process of a founder effect going on. So for example, if you hypothetically envision an island population that's never seen the SARS-2 virus, so almost something like New Zealand because they've had so few cases, but just completely no cases, one person goes there carrying a certain variant of the virus. That virus is going to infect all the people, hypothetically, on the island, and therefore that population is all going to be that one genetic variant. However, the person that came to that island from somewhere else, that somewhere else may have a lot of the other variants. That, in this case, doesn't mean that the virus is more transmissible. It just had this founder effect, where it entered a population where it had lots of people to infect, and therefore lots of cases. Now, it doesn't have to necessarily be an island population to see something like this. It could be that it just hit the right part of a well-populated city, like London. Now, obviously, in London, a lot of people that have already been infected by the SARS-2 virus, so this hypothetical scenario I laid out isn't quite accurate. But this virus, the SARS-2 virus, does have a propensity towards having superspreader events. So a superspreader event is when one person infects significantly more people than would be expected on average. So this is the idea of the R number of a virus. On average, it's expected that one person infects 2 to 3 people, 2 to 4 people, I forget the exact number, but that kind of range. However, in a superspreader event, what's found is that one person is capable of infecting 15 people or 20 people. It's actually thought that these superspreader events might account for up to 80% of the cases of SARS-2. So these superspreader events have a very large impact on the cases of COVID-19. We don't know what's responsible for those superspreader events, unfortunately, because if we did, it'd be a great way to limit the spread of the virus. So it could be down to the virus. Some mutations to the virus are being positively selected for because they can super spread, but maybe not because that virus is going to infect all of those 15, let's say, other people, and they don't all go off and infect 15 other people each, which we'd see much larger explosions. So maybe it's something about the individual, maybe it's the person who is spreading the virus, but we don't know specifically what the host's determining features are. Maybe it's people who have a weakened immune system or something like that, it's unclear. Or maybe it's just one of those really unlucky environmental effects, the right person at the right time, surrounded by the right sort of people who are highly susceptible, or whatever it may be. We don't really understand these superspreader events, but the reason I point out superspreader events and the fact that it seems to be very important for this virus is that just because a new variant, mutant variant of the virus suddenly becomes high propensity, doesn't always instantly mean it's more transmissible. It could have had founder effects through superspreading events because a lot of people become suddenly infected through a superspreader event and then can all pass on that virus. I lay out all of these ideas to give you a framework from which to be critical about any of these kind of stories about mutant viruses and what impacts they have. Just because there's a high incidence of a certain virus mutant doesn't instantly mean that it's more transmissible. More information is required. Now, before we go in to talk about the UK variant, which does actually seem to be more transmissible, I want to talk a little bit more finally about positive selection. So I was talking about neutral selection and things like that, but there can also be positive selection. So in positive selection, a mutation occurs that undergoes natural selection in the way Darwin describes because it gives an advantage to that virus. So a mutant virus versus a non-mutant virus, or technically the wild type virus, the mutant virus has an advantage and therefore it's positively selected and can come to dominance within a population. So if we think specifically about the SARS-2 virus and the population it infects of us humans, there's kind of two main drivers for positive selection. Firstly, there's transmissibility. If indeed mutations occur that make the virus more equipped to transmit from human to human, that will allow it to dominate the non-mutant virus. So hypothetically, let's say that the wild type non-mutated virus infects 2 people. For every 1 person that's infected, they infect 2 other people. So that would spread in a population from 1 to 2 to 4 to 8 and so on and so forth. If we then say, hypothetically, that the mutated virus can infect, for every 1 person, infects 4 people, that goes 1 to 4 to 16 to 64 and so on and so forth. Now, if we have a population, again, hypothetically, of 100 people, where both of those viruses are in that population, so 2 out of those 100 people are infected with these 2 different viruses, the rate of spread by the 5th spread, the mutant virus has completely dominated that population. So the numbers going 1 to 2 to 4 to 8, while the other one is going 1 to 4 to 16 to 64. At that stage, 72 people have been infected, plus the original 2, and 90%, 64 of them, are the mutant virus. And since I've only laid out a population of 100 people, the next spread, all the people will be infected, and 90% of them are going to be from this mutated virus. So this is how mutations that enhance transmission can allow a virus to dominate within a population. So that's, of course, a hypothetical situation, as I say. That's not what's going on with this new virus. That's not what I'm saying is going on with this new mutant virus, but it's an example of how a mutated virus that can transmit faster may come to dominance within a population. Another potential positive selection that could occur on the SARS-2 virus in the human population, though, is the ability to escape from immunity. So again, if we take a hypothetical 100-person population and 90 of those people have immunity to being infected, this means only 10 people can be infected. However, if a virus mutates and a new variant emerges that can infect people that were infected with the original virus, therefore nullifying the immunity they had, that virus has 100 people it can infect, whereas the non-mutant virus has only 10. Therefore, the mutant virus is going to go on, and even if it infects 2 people, that 1, 2, 4, 8, it's going to come to dominance because it has a full population it can target. Again, this is just hypothetically laying out the kind of situations of how viruses might mutate and evolve and how they could therefore come to dominance within a population, which is what we're seeing with this UK variant. And that finally brings me on to talking about that. So this variant virus identified in the UK is officially designated B.1.1.7, so that's B.1.1.7, so there's a dot in between each part of it. This virus has 23 mutations in total compared to the main circulating strain, so this idea of the non-mutant versus the mutant, there are 23 mutations. However, only 17 of those are synonymous changes. So again, back to the top of the video where I was talking about synonymous changes are the ones that change proteins, and non-synonymous are ones that have no impact on the protein. So that is quite a lot of changes compared to the other viruses. And of most interest right now are the fact that there are 8 mutations within the spike gene. So this is the one that everyone is thinking about right now with regards to transmission and vaccines and immunity. So the spike, as always, is the bit on the virus that sticks out, and it's how a virus binds to and infects a cell, key in a lock, as a metaphor I often use, and it's also the target of antibodies. So antibodies bind to the spike and stop that key going into the lock, and therefore the virus can't infect cells. So there's 8 mutations to spike, which potentially could impact transmissibility, so the ability of the virus to bind to and infect cells, and the ability of antibodies to bind to and inhibit that process. So both the ideas I was discussing for positive selection. Now, there's a couple of interesting mutations in there. So one of those mutations is one that's been previously seen, and it's referred to as N501Y. So this is just how mutations are designated. What that means is that the amino acid asparagine, so N, at position 501, the spike gene, has been converted into a tyrosine by mutation. So tyrosine is Y. So the DNA changed, and that resulted in the amino acid that makes the protein changing from an asparagine into a tyrosine. Why this position is very important and interesting is because 501 is part of the receptor binding domain. So that's part of the spike protein that actually directly interfaces with the part of our cells that the virus uses to infect, which is the receptor called ACE2. So it's kind of like the teeth on the key, sort of the most important part for actually doing the function of the key. Now, why the 501 mutation is particularly important is we've seen it, or particularly interesting, is we've seen it in other viruses that are out there in nature circulating. So this UK variant is a very specific variant with these 23 mutations, 17 that change proteins, 8 of those in spike. But we have seen other viruses that carry N501Y, and they've been assessed in the lab. So people have found that N501Y, so a virus that has Y instead of N, combined better to the ACE2 receptor, and that seems to enhance the ability of that virus to infect cells in the lab. However, importantly, that doesn't seem to impact the ability of antibodies taken from people who've had COVID-19 doesn't stop the ability of those antibodies to inhibit infection. So if you take serum from patients and use that, you find that that is equally effective at inhibiting a virus with an N at 501 as a Y at 501, even though the Y seems to enhance the ability of that virus to infect cells. So it looks like that mutation is capable of enhancing transmission, potentially, but doesn't impact the immunity, so natural immunity from people, and by extension, probably vaccine-based immunity. However, that is a very specific case of looking at that one mutation in isolation. And as I say, there's 8 mutations in this UK variant that are defining this variant in the spike gene. So that hasn't been assessed yet. It's too early. People haven't been able to do the experiments yet to work out if in the lab there are impacts to the ability to bind to ACE2 and the ability to infect cells for this specific variant. That will take a week, maybe more, 2 weeks, being that we've known about this for a little while. People have grown up the virus. We were calculating it in the lab today that if we hypothetically got it, we reckon we could have some data within 2 to 2.5 weeks or so. But right now we don't know if this variant is better at infecting cells. The reason it's being said that it's more transmissible is because of the growth in the population we've seen. So we've known about this virus since about September, and it seems to be at a prevalence of about 60% of new infections in London. So the growth of this, using that information combined with the genetic information about how this virus evolves and the epidemiological data, it really suggests this virus is more transmissible. However, that is still requiring lab-based confirmation, but the data we have does indeed suggest it's more transmissible. Now, a mutation in this variant strain that is of a little bit more concern, potentially, is a deletion of 2 amino acids. So deletion at position 69 and 70 of a histidine and a valine. The reason that's a bit more concerning, potentially, is because that type of mutation, again, has been found out in wild transmitting viruses, not in conjunction with all the other mutations of the B.1.1.7 virus. But that has been suggested in the lab to be less sensitive to antibodies from patients. Again, though, I want to stress that with regards to this specific circulating variant, we don't know what impact all of the mutations are going to have on the ability of antibodies from patients to inhibit the virus. We still need to do those experiments. As of the 23rd of December, I've seen no data that can tell me one way or another whether this virus is going to escape from immunity from natural infection. There's not been any mass spread within people being reinfected of this virus. There's not an overrepresentation of people who are being reinfected, having already been infected by SARS-2 one type and being infected by this variant. So potentially it's not escaping immunity. But right now we don't know. So potentially something concerning there, but really too hard to say with any conviction and too early. As of the 23rd of December, I've seen no assessments of what impact these mutations have on this virus outside of the genetic information and the number of the people that have this virus. So right now it's up to about 60% of new cases in London who have this virus. Importantly, even though there's this huge uptick of the virus, this variant, there hasn't been a huge surge in severe cases. So it doesn't look like it's causing any more severe disease. Now, obviously, I was just talking about natural immunity and escape from the antibodies collected from patients, and that leads me into talking about vaccines. So again, right now, it's far too early to say whether this variant strain is going to have any impact on the vaccines that are now being rolled out. So from Pfizer and BioNTech and Moderna, which are both approved for usage here in the US. In the UK, it's just the Pfizer vaccine. So the predictions of what impact these mutations are going to have on the structure of the spike protein don't suggest anything that is going to impact immunity. But at the minute, it's all just predictions. Pfizer and BioNTech have come out to say that they think within 2 weeks they'll have information about whether serum taken from the people they've vaccinated is still capable of inhibiting infection by this variant virus at the same rate. So right now, as of 23rd of December, all I can say is it's too early to make any conclusions, but there is optimism that these mutations aren't going to impact the efficacy of these vaccines, but only time can really tell because we need the time to do the experiments. So just sort of as the summary there, it does seem like this virus is transmitting better based on how rapidly it's come to dominate within the population in the southeast of England and particularly in London. But that is still just based on genetic and epidemiological data. We haven't got any lab-based data to show that. We also don't yet know what impact these mutations have on immunity, be that natural or vaccine-derived. As of right now, there's no reason to think that there's going to be a massive escape from either of those based on the mutations. Maybe some loss of natural immunity, but not a complete loss. And maybe there'll be some lowering of efficacy of the vaccines, but these vaccines seem to be incredibly effective at 90+% efficacy for protection. So even if there's a small drop off, they're still going to be very, very good vaccines. But that really is just speculative right now. We don't know, and we need to do the experiments, and I'm sure they're ongoing, and I look forward to seeing the data that comes from Pfizer and BioNTech and other people. But an important point is that all the things that we're trying to do to limit the spread of the wild type coronavirus, the non-mutant strains, variants, sorry, there's not new strains, the non-mutant variants, they're still effective against this virus. Wearing a mask, washing your hands, physically distancing and being in well-ventilated spaces are still going to reduce the spread of a more transmissible virus. So we still have to keep doing those things. If we were doing those things more effectively than we are in certain areas, there wouldn't be the emergence of these new variant mutated viruses. The virus can only mutate when it infects people. So if we can actually lower the amount of infection, we'll see fewer mutated viruses. So these are the best ways to block spread of this virus. So please continue to do all those things. So finally, just before I close out, I want to say why the UK. So I'm based in Baltimore, but I'm from England, as the accent may tell you. This is the UK variant, and the reason is because the UK is doing an incredible amount of genome sequencing of SARS-2 viruses collected from patients. I saw a tweet recently that suggested it was doing 100 times the amount of genome sequencing as America, and the UK is a lot smaller than America. I haven't been able to work out where those numbers came from, so I don't necessarily take that for rote, but the UK is doing a huge amount of sequencing. So of course, it's going to find when these genetic variants emerge. And I think that's really important, and everywhere needs to be doing that. Viruses mutate incredibly rapidly, as I'm saying, and there is the selection pressure to keep spreading within a population, be that by overcoming immunity or increasing transmissibility. Again, none of that necessarily impacts how deadly the virus is, but the virus will evolve to continue to spread. That is the selection pressure on the virus. So we need to be aware of if these viruses emerge, and we need to be aware rapidly whether they are escaping immunity. If we find that variants are emerging that are escaping the vaccines, we need to know that as soon as possible. So I think it's really great that the UK are doing so much sequencing, such as to find this variant. I have no doubt this variant is in America already. It just hasn't been seen because there haven't been enough sequencing of the viruses that are infecting people here. I think it's also important that scientists, we scientists, start to bank serum samples from patients who've been infected and from patients who are receiving the vaccines, because that's how we can quickly test if indeed there is any escape from the antibodies that are being induced in those people. We want to know within a week or two if these new mutant viruses are escaping the vaccine. In principle, these mRNA vaccines are super easy to change. Should indeed a new spike emerge, you can change the RNA in the vaccines very easily. I don't know the regulatory functions there. I don't know how easy it is to go through the regulatory loops, but scientifically and experimentally, it's very easy to make the new variant as the vaccine. So we can respond rapidly if we know that these viruses are mutating and what they've mutated into. So just a word to say that's why it came from the UK and why the UK is doing a great job in finding these viruses. Obviously, it'd be great if there weren't these viruses spreading. If there were no cases, we'd be in a much better situation, of course. But since the virus is here, we need to be finding out what variants there are. So all that said, thank you for watching, as always. I've done quite a long video on this tonight because it's an important topic that I really want to stress in that viruses are constantly mutating and they evolve very rapidly. Natural selection is in hyperspeed for viruses because of how quickly they mutate. So we're going to see stories like this. Eventually, I'm sure we'll see a story of a virus that has mutated and is no longer impacted as effectively by the vaccines. We need to be in a place to find those variants rapidly, and that's why I wanted to stress that the UK finding this is actually really good and really important. But we will continue to see stories of viruses mutating, and that's why, as I say, I wanted to put out a long video. The best way to stop viruses mutating, though, is to stop people being infected, and all the measures, such as wearing a face mask, washing hands, keeping physical distance, staying in well-ventilated spaces, they all work. Regardless of how well a virus transmits between people, having all those layers of protection will still block that virus's ability to spread. So with that said, Merry Christmas, Happy Holidays, Happy End of the Year, whatever it may be. Please stay safe in this period. And please be sensible with all the things. Thank you for watching. Drop a like if you found it useful. Ask questions, comments below, all those kind of things. I'll probably take a little break from doing these videos over this period, have a little bit of rest. But I'll be back in the new year, if not before. So, thank you.